I'm Sandhya. I'm studying Department of Microbiology. Now we are going to discuss about the topic is antigens. First one is introduction. A substance that can induce an specific immune response is called as an antigen. It can react specifically with antibodies or sensitized cells. The ability of an antigen, it will produce an immune response and it will react the products. It's called as antigenicity. The ability of a material which one is induce a specific immune response is refers to it as a immunogenicity. They are known as the immunogens. Another one is structure of antigens. The antigens has a binding site is called epitope or antigenic determinant. The antigen binds to the antibody or any surface receptor with the help of epitope. The binding site of antibody is called as paratope. As antigen has many epitope, an antigen with many epitope is called as a multivalent antigen. The epitope of antigen may be different types. They are bind with the many types of epitopes on the surface is called as a surface epitopes. The surface epitopes are functional and the hidden epitopes are non-functional. There are two types of function in the surface epitopes. The epitope is made up of usually 6 to 8 amino acid and glucose units in a polysaccharide antigen. Another one is structure of antigen. This is the structure of antigen, which are first they are made up of two chains. One is light chain and heavy chain, and made up of two region. One is variable region, another one is constant region. And in that regions, they are binding sites. Antigen binding sites are visible in the light chain and heavy chain. They are binding together. Another one is chemical nature of antigen. Proteins, nucleoproteins, nucleic acid, polypeptides, lipoprotein. These are the chemical nature of antigens. Essential factor for antigenicity. It's being very large molecular size. It's usually in protein nature. The proteins are more antigenic than polysaccharide. Soluble molecules are more antigenic than insoluble molecules. The antigens must be degraded into fragments. Antigen antibody binding. The binding of antigen and antibody is specific. The binding can be compared to a lock and key. The epitope of antigen bind to a paratope of antibody. The binding occurs through the covalent bonds. Antigen antibody binding means they can bind with each other with the specific paratope or epitope. This is called as an antigen antibody binding. Another one is types of antigen. Exogenous, endogenous, thymus independent, thymus dependent antigens, apton cross-reactive antigen. First one is exogenous antigen. The antigens entering the body from outside. When the antigen entering from the outside is called as a exo. Exo means outside. Exo means outer region. That means the foreign antigen. Example, bacteria, virus, fungi, and many things. The foreign particles are entering is called as exogenous antigen. Next one is endogenous. The antigen are derived from the body. That means endo means inner, exo means outer. Derived from the body is called as endogenous antigen. For example, nucleic acid, MHC complex, etc. They are classified into two types. One is autoantigen, another one is alloantigen. Auto means they will present from the own body. Auto means own, self, it will produce an antigen. This is called as an autoantigen. Example, nucleic acid and polypeptides, etc. we will say. Another one is all alloantigens. The antigen present in different members but in the same species is called as the allo. Allo means different. This is called as they are present in the different members. They have a genetic polymorphism with the individuals of same species. This is called as alloantigens. Another one is thymus independent antigens. Thymus independent antigens are non-proteins. This is the structure of thymus independent antigen. First, the B cell bind with the microbe. It will produce antibodies for a specific receptor and it will bind to the NK cell and finally it will process and will the phagocytosis. Another one is thymus dependent antigen. The MSC peptides and TCR interaction. How the major histocompatibility complex peptide and the TCR reaction or interact with each other. Thus the thymus dependent antigen, B cell dependent antigen. One is B cell, another one is T cell. They are bind with each other with the MHC class 2 molecules and peptide antigen. With they are stimulating some receptor. One is CD40 in B cell, another one is CD40L in T cell. This is a thymus dependent antigen function. It will works. It's a mechanism. Another one is aptan. Aptan is called as incomplete antigen. Why the aptan is called as incomplete antigen? Because they combined with antibody to initiate some immune response. They will stimulating the immune response. It's called as an incomplete antigen. That's why we saying it's not being immunogenic. Aptan become immunogenic on combining with large molecule. It's when it's become immunogenic, it will combine with large molecules. For example, the chemical nature is dinitrophenol. Another one is cross reactive antigen. What is the cross reactive antigens? That means the antigen antibody, they will cross react with each other with a specific. It will determine by the partial configuration of the antigenic. It will a determinant group, but the antigen specificity is not stable. It will cross react between the antigen or stereochemical similarities. One such cross reactive antigen is heterophile antigen. This heterophile antigen is what discovered by Werner Forssmann. Thank you.